Hello, everyone. In this chapter, we are going to explore life and dive in science, which is basically in relation to life. How do we acquire knowledge in relation to biology, right? As we address this topic, I would like you to focus on few questions. One, what is biology? Two, what are the characteristics found in living systems? What do we call life? How do we classify living organisms? Where in the big picture do humans fit? Because remember, this course, it's about human biology, right? And then we are going to address the question about how we acquire knowledge in biology using a scientific approach. What is that approach? And finally, as we gather all this information using a scientific approach, question is, where do we publish that information to communicate it to people, right? Where do we obtain the information to be aware of scientific discoveries? I'm talking about accurate information, right? So these are questions I would like you to keep in mind as we cover this chapter. Our first step is going to be on the characteristics of life. When do we say that something is alive? What is specific to life? Well, life, first and foremost, is very organized. It's organized such that we can find out information about its organization from the smallest component that is found in a living system to the largest component. The smallest component being atoms. Atoms come together to form molecules. The largest component in living system being the biosphere, which is the global ecosystems in the universe. Other characteristics of living system, it's that living systems are able to use raw material, extract energy out of that raw material, and that raw material is obtained from the environment. Living organisms have to be able to maintain a relatively stable internal environment, a characteristics that we call homeostasis. Also, living systems have to be able to respond to stimuli, whether the stimuli are internal or external. They have to be able to respond. They have to be able to adapt to this environment. Then living organisms have to be able to reproduce, leave behind offspring, and this offspring themselves have to be able to reproduce. Living organisms have to be able to grow. And finally, but not least, all living systems, regardless, have an evolutionary history. In other words, in a given environment, living systems are going to be able to adapt to that environment in a long-term fashion. We're talking about years. We're talking about centuries, right? When we look at living organisms and we look at the different levels of organization of life, we can look at life on a size scale basis from the very smallest component to the largest component. The largest component, the most inclusive part of living systems being biosphere. Biosphere, it's everywhere life is found in the universe. When we zoom in the biosphere, we're going to find a smaller structure that we call ecosystems. Ecosystems are going to be a mix of living components and non-living components interacting with each other. Remember that living organisms have to acquire raw material from their environment, right? That raw material can be water, right? And other materials. So in an ecosystem, we have living component and non-living component and how they interact with each other. Then if we zoom in ecosystem, focusing on life, we are going to have communities. Communities, for instance, on this picture right here, you have a tree, you have grass, you have humans. These are different types of organisms, different species living in the same environment. Now we are going to call this community, right? If we look here in a savanna, we are going to have zebras, we're going to have grass, we're going to have trees. This is another community. Zooming in the community, we are going to find populations. Populations, in this particular instance right here, we are going to zoom in the human. That's one species of organism. Groups of many humans constitute a population. Zooming in the population, we are going to find organisms. This is one organism, a human. Within a human, we are going to have organ systems. An example of organ system is going to be the nervous system. In the organ system, we are going to find many organs. For instance, in the nervous system, the brain is an organ. Organs themselves are going to be made up of tissues. An example of tissues in the brain is the nervous tissue. And tissues are going to be made up of cells that are similar and come together to form, to work toward the same function. Now, in the nervous tissue, a specific cell is going to be the nerve cell. A nerve cell is going to be made up of complex molecules, the macromolecules, including carbohydrates, proteins, nucleic acids, lipids, right? All these molecules are going to cause cells to exist. And the molecules themselves exist because of an assembly of atoms. So now, if we look at this organization of living things, the most inclusive structural level is going to be the biosphere, while the most exclusive structural level is going to be the atoms. So I expect you to know, when given a number of levels of organization, which one is the most inclusive and which one is the most exclusive. Now, as we consider life, we have to be able to classify living organisms. And the reason why we have to be able to classify living organisms, it's because by knowing how living organisms are connected, we can actually solve a lot of our problems, right? For instance, treat diseases. That's an example of problem that we are going to be able to solve by knowing how living organisms are classified. Anyway, living organisms are classified in domain, kingdoms, phylum, class, order, family, genus, and species. if we consider, for instance, a human and a house cat, these two organisms belong to the domain of eukarya. These are eukaryotic organisms. These two organisms fall in the same kingdom of Animalia. They are both animals. A house cat and a human, they are both animals. We also notice that these two organisms, they both have a spinal cord. They fall in the phylum of Chordata. And they both have hair. A house cat and a human, therefore they fall, both of them, in the class of mammals. Now, we are going to start seeing some differences when we dive in the order. In the order, humans are primates, while house cats are carnivora. In families, we are going to see further differences. Humans are part of the Huminidae family, while house cats are part of the Felidae. Human belongs to the genus of Homo, while house cat belongs to the genus Felis. Human belong to the species Sapiens, while house cat belongs to the species Domesticus. So we see, looking at how we classify living organisms, that at some point we have similarities, but at some level we also have a lot of differences, right? Now, with that being said, because this is human biology, what is characteristic to human and how humans are different from other living organisms? One, humans have this particularity that they can pass down a cultural heritage from parents to offspring, from generation to generation. Humans have a highly developed brain. They have a complete upright stance. Humans have a creative language ability. They are able to use and create tools, and they are able to modify their environment in a tremendous manner, modify the environment to their own benefit. But at the same time, though, humans have to be aware of sustainability as they transform their environment. That is the reason why in this course, human biology, we are not going to only dive exclusively in the organ systems. We are also going to look into our environment, the biosphere, and how we interact with that environment, what kind of impact we have in that environment, and how to create sustainability. So, as we study life, we are using a scientific approach to develop knowledge about living organisms. That scientific approach, it's a systematic way to approaching questions that we have about life. So, science, it's a way of knowing about living systems and about the nature around us. What is characteristics of science is that science is objective. we have to be able to measure. We have to be able to quantify. It's not about what we believe or what we trust. It's about what we can see, what we can measure, right? What we can quantify. Science allows us to reach conclusions, and this conclusion may change over time based on what we know at that time, based on the tools we have, right? Now, because of technology today, we are able to modify some of the knowledge we had and say we thought it was this way, now we know it is different, right? So we adjust things as we discover more and more things in science. So we are going to use a scientific method to acquire knowledge. Because of the scientific approach, we have acquired a tremendous amount of information about life. For instance, we know that all living organisms are made up of cells. That's one. Two, we know that cells come from pre-existing cells. And finally, we know that a cell is the basic unit of life, meaning that the cell is the smallest component in a living system that has all the characteristics of life. Using a scientific approach, we know that all living systems have to create a relatively internal, stable environment, right? What we call homeostasis. We know because of science that genes are the units of expression that produce features that are measurable, that are quantifiable. You look the way you look because of genes, right? Because of science, we know that ecosystems are going to be made up of living organisms and non-living components. They interact with each other, and the environment can influence living organisms in an ecosystem. And finally, we've been able to understand that living organisms are able to evolve over time, so that today we know that all living organisms have a common ancestor, okay? But each organism have adapted differently based on the features they have to adapt to that environment, because not all organisms have been able to adapt. Those that have not been able to adapt have disappeared altogether. Come on. Are we serious here? There we go. So, what is the scientific approach that we use to find out information? Well, there are a number of steps that we are going to follow to find out some specific information. A scientific approach always starts with observation. That observation is going to trigger questions. These questions are going to cause us to think about potential explanation or hypothesis. We can have many hypotheses for a particular question, right? As we draw this hypothesis, which are tentative explanation, we are going to think, well, how can we verify that hypothesis? Then we have to design experiments, right? And as we design experiments, we know that it's going to cost us something to execute this experiment. It's going to take us money, it's going to take us time, it's going to take us material. So that before we execute an experiment, it is good to have a good rationale before running that experiment, so that we have to have a prediction. That prediction is going to be a way for us to know, to come with potential outcome if we execute that experiment. That way, we are going to likely save time, money, and energy. So it's good to have a prediction. Once we have that prediction, then we are going to execute our experiment. We are going to run tests, right? So we're going to run some experiments. And as we run experiments, we're going to gather results. And this result, we're going to analyze them and draw conclusions. This conclusion may support our hypothesis or may not support our hypothesis. If our conclusion support our hypothesis, then we are going to say that, well, our hypothesis is correct. If the experiment do not support our hypothesis, then we are going to reject that hypothesis, right? So, one good example of a question we've been able to solve in science, it's actually in biology. It's what causes stomach ulcer in human. For a long time, this was a question that didn't have an answer. Speculation was that stomach ulcer is caused by witchcraft, or some weird situation, maybe someone committed a sin. But today, we know that stomach ulcer is caused by a bacterium. But how did we come to that conclusion? First, the scientist who did the work on that particular aspect made, first of all, the observation that many patients had a particular bacterium near their ulcers. So the scientist, actually he was an Australian, he made a hypothesis that, well, it is possible that a bacterium called Helicobacter pylori is responsible for the production of stomach ulcer in human. And actually there is a funny story about that. This guy, actually, to prove that Helicobacter pylori was responsible for human ulcer, he actually drink a mix of Helicobacter pylori and became crazily sick. But eventually he didn't die, but he experienced some major discomfort. Anyway, let's close that parenthesis. So what the scientist did is to run some experiment, right? After making the observation, asking the question, right? He ran some experiment. So in the first experiment, he isolated Helicobacter pylori in stomach ulcer from patients. In the second experiment, he showed that when swallowing Helicobacter pylori solution, people could develop inflammations in their stomach. Actually, he himself did that. He swallowed Helicobacter pylori solution, which was crazy. But, you know, scientists can be very crazy sometimes. And then he reached the conclusion that Helicobacter pylori was the causative agent of stomach ulcer. And that because of that, actually, Helicobacter pylori, the stomach ulcer, can be treated using antibiotics. But when we run antibiotics, question is, among two antibiotics, which one is good at treating stomach ulcer? Or between two good antibiotics, which one is better? So to find that, scientists are going to work with group of people. Say we have a group of people with stomach ulcer, right? We are going to prescribe to this group of people antibiotics. This antibiotic is going to be the experimental variable, right? So now, one term we are introducing here and that I want you to remember is the concept of variable. Here we have the experimental variable, which is the variable that is intentionally changed in an experiment. In this particular case, the variable is going to be the antimicrobial, the antibiotic, right? All other variables in the experiment are going to remain constant. So that's one. Two, now we are going to work with groups of patients, that are treated with what we know to be the variable, the antimicrobial, and groups of patients that are treated with what we know to not be the variable, a placebo, for instance, right? So a test group, which is a term I want you to know, a test group is a group of subjects that are exposed to the experimental variable, in this particular case, the antimicrobial. A control group is a group that is not exposed to the experimental variable, and this group is used only for comparison to see if our antimicrobial is actually effective at treating the disease or not. So here we have a group of 9 people. These 9 people were further divided into 3 subgroups. The 9 people have stomach ulcer, and we are going to give them a substance, and we are going to see which substance is good at treating the disease or not. In the first group, we're going to give that group a placebo, so a chemical that technically is not going to do anything to the stomach ulcer. Test group 1 is going to have antibiotic A. Test group 2 is going to take antibiotic B. Then we are going to follow this patient over time, and then we are going to gather data. Did the molecule they took affected their health, yes or no? If so, which proportion? So here we have the data. We see that the control group, the group that received a placebo, only 10% of the control group saw some effect after taking the placebo, which actually is almost nothing, right? In the test group 1, we have that 60% of the subject saw an improvement in their health, while test group 2 showed a better improvement. While in test group 1, we have 60% of patients that showed an improvement in their health. In test group 2, 80% of the people saw an improvement in their health, showing that antibiotic B is more effective than antibiotic A. Therefore, when designing a treatment to treat stomach ulcer, patient will be most likely prescribed antibiotic B rather than antibiotic A. And also, we show that a treatment is essential for improvement of health when someone has stomach ulcer, because the placebo versus antibiotic, we see a huge difference. So once we gather all this information, question is, what do we do with that information? One, we have to communicate that information. We communicate that information through scientific journals. The scientific journals are going to be the best source of information. However, the major issue with the scientific journals is that they are very difficult to read. They are very difficult to read. It's going to be difficult for the layperson to understand the message in a scientific article. So often the layperson is going to benefit from reading a secondary source of information, which is that someone has to first read a scientific journal, and then they're going to summarize the information in a way that is palatable to the layperson, right? But the problem with that is that the person who is actually making the information in a manner that is easy for the layperson to understand, they can make mistakes in interpreting the scientific information, which causes a lot of confusion in the population. So we have to be careful where we are obtaining our information from. So it's going to be very important, for instance, when we go online, to pay close attention to the website we are visiting. What is the URL that we are visiting? So URL with extension edu are trustworthy. .gov and .org or .org, they are all extensions that are acceptable, right, or trustworthy, while .com is not trustworthy, .net is not trustworthy. Now, I'm not saying that extension.com cannot provide you accurate information. Yes, they can, except that in a scholar environment, in an academic environment, we are not going to use the sources as trustworthy sources, even though they may contain accurate information. So we have to be cautious about where we obtain information and what we do with that information. We have to be aware of the fact that some source of information can actually provide information on anecdotal data. Okay, so we have to be aware of that. We have to understand the methodology and the results as we read the information. Does the data justify the conclusion? So when we read we have to read with a critical mind, right? We have to be able to read graphs to understand what these graphs are conveying as information, and we have to be able to understand the statistics because statistics plays a very important role, right? We can say that a particular drug works on people, but the question is how many people in the population were tested. And even when we consider the number of people, are we also consider where the people come from? Right? All these aspects are going to be very important. We know that because of all the knowledge that we are acquiring, we are developing new technology. So technology is the application of scientific knowledge for the good of human. However, we have to be aware of the downside of applying technology, which raises questions of bioethics. What is bioethics? Basically, it's the branch of ethics that is concerned with the development and the consequences of technology in living systems, right? Today, we use very much what we call genetic engineering, for instance, to design genetically modified organisms, right? When we talk about genetically modified organism, GMO, sometimes this can cause fear in us. This can cause controversy, right? Now, let me take you some examples. We have, when we take a drug such as human insulin, right? You go to the pharmacy, you go get human insulin, right? The question I'm going to ask you is, where does that human insulin come from? How do scientists do to produce that human insulin? Well, know that bacteria are genetically modified to produce human insulin. We never question human insulin when we go to the pharmacy and get it. However, on the other hand, when it comes to food and genetically modified organism, we think, whoa, we don't know what is in this food. Maybe we're going to get sick. What is going to happen with this genetically modified food in our system? Now, we are right in asking the question, but we have to be cautious on where we obtain the information, and what we have to be cautious about misleading information. All right? So, genetic engineering raises a lot of questions, a lot of controversies, right? But we have to know that it's both for the good, but we also have to be aware of bad things that can happen with genetic engineering. Therefore, we have to apply bioethics. Then we have the threat to biodiversity. Yes, the fact that we are producing genetically modified crop is a major threat to biodiversity because we tend to favor that genetically modified crop. For instance, in the United States today, it is very difficult, if not impossible, to find corn that is a wild type corn. All the corn that is planted, that is used in the United States these days, is genetically modified corn. Anyway, so we human are able to influence our ecosystem, our environment for our good, but we also have to be aware of the fact of the impact of our activity on our environment. Today, we see emerging diseases. These emerging diseases sometimes occur because of our impact on our environment. We talk very much about global warming today, and we think that carbon dioxide, a huge production of carbon dioxide, causes increase in the global temperature. Some people are going to argue that this is not true. It's only cyclic and it has always happened in the environment. Well, some of it is true, some of it is not true, right? We have never been in the history of the universe a time where we have produced this much carbon dioxide. Anyway, let me close that parenthesis. So this actually conclude this chapter. Thank you for your attention. See you in the next chapter.